Good afternoon, everybody. My name is Cory Inman. I'm an assistant professor at the University of Utah in the Department of Psychology and in the neuroscience program. Today, I'm excited to tell you about our lab's work that we playfully call Cog Go. This is a play, of course, on Pokemon Go with the idea that we want to take cognitive neuroscience and how we do cognitive neuroscience from the laboratory out into the real world in the same way that we've taken video games from on a computer screen out into the real world with location-based augmented reality games like Pokemon Go. And specifically, I'll be telling you about our work that is aiming towards being able to capture and eventually one day hope enhance episodic memories that are made in the wild, complex, naturalistic, rich, real world. So I'm very excited to tell you about this work, and I'm happy to answer any questions that people email me after this talk. And I really appreciate LabRoots for giving me the opportunity to present this work. So I first want to start just by saying and acknowledging all of the wonderful people that I am just here to represent that are actually doing this work and spending their efforts to make this work possible. From my lab, the Immersive Neuromodulation and Neuroimaging Lab at the University of Utah, and my collaborators like Luis Garcia and Tyler Davis. My postdoctoral lab at UCLA in Dr. Nanthi Suthana's lab, who really allowed me to learn how to do this work. And then all of my collaborators during my PhD and my first postdoc that helped me learn how to do human electrophysiology and really allowed me to pursue these types of ideas in my career. So I want to start with just kind of framing, the sort of approach that my lab tends to take. I think it fits really well with the theme of this LabRoots sort of sub theme. And that is that, currently, I think we have done a great job in neuroscience, maximizing the opportunities that we've had to do neuroscience with humans and with animals. And often these studies, for various technical reasons, have had to be fairly static in nature, and that's partially to gain experimental control over the experiments that we're doing, or to minimize any sort of artifacts that might come about from movement. But I would like to also point out that this is not exactly how we experience the world. That we experience the world, and we think and behave in the world in a much more dynamic sense than we're often able to capture in the real world or in the laboratory. And so, each of these first-person videos is, just here to kind of point out that this is how we experience the world from this egocentric point of view, moving throughout the world. We're kind of our own agents and own directors that are directing our actions and behaviors. And that ultimately, I think what we want to understand in neuroscience is how we do this, how we do these things, whether it's hiking through the woods or playing a sport, the city, and navigating around the world. This is ultimately what we want to understand. And so, I was mentioned some of the technical limitations, doing any sort of brain recordings in freely moving humans has been really challenging due to technical limitations, from functional MRI to MEG, scalp EEG, even intracranial EEG. They all have these traditional methods. They require immobility to minimize movement artifacts, or in the case of fMRI, they're recording at slower temporal resolutions, or scalp EEG. It's often really primarily recording from more superficial brain areas. And even with intracranial EEG, it's not, you can move around with intracranial EEG, except you're actually tethered to a amplifier with a few wires coming out of your brain. So you're literally sort of tethered to the wall by your head. So you can only go so far in terms of movement. And what I'll tell you about today is some opportunities that we've recently been able to take advantage of to record from the brain as people move throughout the real world. And all of these traditional methods have been able to tell us a tremendous amount about how the brain works, and we've learned a lot. But I think that there are technical opportunities, that allow us to start pushing the boundaries of how we do cognitive neuroscience. And often these traditional methods, because of these limitations, we've had to use these fairly unnatural behavioral tasks that are low in ecological validity. So, when we ask ourselves in my lab, why should we be spending all this time working towards understanding cognition and emotion and other types of behavior in the wild, is that one, as I mentioned before, I think this is ultimately the type of neurocognitive behavior that we want to be able to explain in humans. Many of these abilities are fundamentally human abilities, like the ability to describe our emotional experiences, or to remember deep into our past, or imagine far into our future based off of our past experiences. There's, of course, the current limitations of non-invasive neuro. And finally, related to some other work where we do amygdala stimulation to enhance memory in our lab, in these really controlled settings. One of the things I realized as I was doing that work is that if we ever want to realize the promise of neuromodulation, say for memory enhancement, or even for things like post-traumatic stress disorder or other types of sort of psychiatric disorders that come at particular moments in time, it's going to require us to have some understanding how to do experiments in the dynamic, complex, real world. And so my laboratory starts by, of course, acknowledging that learn from doing laboratory experiments where we have high experimental control, and we relinquish some of the ecological validity to that experimental control, doing tasks on a computer, sitting in a bed with our intracranial EEG patients, or with undergraduate participants. But ultimately, I think we want to move towards having lower experimental control and higher ecological validity. And we can do that through maintaining some of this experimental control with virtual reality and augmented reality, but also trying to gain some ability to have more naturalistic, dynamic experiences in patients. And so ultimately want to move from the laboratory out to the real world. But the more I started to think about what we really need to try to understand at this point, is that we don't really have any data from people that are actually doing real world behaviors as they move through the real world, where we have brain recordings. And so I think there's a really great opportunity even now, without taking the kind of incremental steps from the laboratory through virtual reality to augmented reality and then to the real world, to actually do some more observatory observation-based experiments or exploratory experiments in the natural environment, with people doing tasks that we all do on a day-to-day basis, like navigating through our environment and trying to remember how we got from one place to the next. So there's a really great opportunity to have some of that actually feed back on the sort of more controlled studies that we can do in the laboratory, to try to inform what relationships might matter actually out there in the real world. I think it's also dangerous to only stick with doing kind of hypothesis testing with highly controlled experiments in the real world, or in these kind of constrained environments, because it's likely the case that you might actually not end up explaining how the brain works. So today I'll tell you a little bit about recording from the brain in the wild, and mention some ideas that we have for what we might be able to do by doing neuromodulation in the real world as well. So we call this our Abbey Road picture here, and this is our setup that we call the photon pack. So first, I'd like to start by saying that there's lots of new opportunities for doing cognitive neuroscience in the wild. There's motion tracking that are used in Marvel movies, as people wear accelerometers that give you a biophysical model of how your body's moving through space. There's wearable eye tracking that just records all of the data to a phone and then uploads all that data to the cloud. There's these high quality cameras that have gone through many generations at this point. We all have really great GPS on our phones that can tell us where we are at any given point in time. And we can even maintain some control in the real world by presenting things with things like augmented reality. So that's what I was showing over here with these butterflies kind of just floating in the room. And there's even the Apple Vision Pro that gives you these types of opportunities now as well. One of the first examples of this was Pokemon Go, which is why we start this talk by talking about Neuro Go. And that was where you actually were able to put Pokemon in your real world, and you would be able to catch them in your real world, and you get a real experience out of that, and it really kind of took the world by storm when it came out. But what really opens the door is not just all of these wearable kind of sensors that you can wear on your body or presentation displays, but really what gives us this ability to move into the real world is these novel patient populations that have implanted neural recording devices that record on a regular basis. And so this is known as the NeuroPace Responsive Neurostimulation System. This was approved in 2013 and is implanted in over 2,000 people at this point, probably even closer to 2,500 or even 3,000 people at this point. This is used to help treat a patient's epilepsy. So the electrodes are implanted into parts of the brain that have been shown to provoke the patient's seizures and for the place from which the patient's seizures spread. And so what it does in its normal operation is it listens for seizures. And then when it detects something that looks like a seizure that might start, it stimulates the brain in order to interrupt that activity and to from getting synchronized with other parts of the brain and spreading. And what we've shown in what Dr. Sathana has shown in a couple of her earliest papers on this work is that, one, we get lots of patients that have things like hippocampal recordings or cingulate recordings, amygdala recordings, things like that. And that when you record from these electrodes, even though they're in seizure onset zones, that you actually get pretty reasonable data that meshes well with what we see in animal studies, like theta oscillations in the human hippocampus that are modulated by how fast someone moves through the environment. So this has been done in virtual reality just by having patients walk around an instrumented room. Now, instrumented rooms are really great. They allow you to do a lot, but you're still constrained to that smaller environment. And so one of the things that I wanted to do when I was a postdoc was try to figure out and develop a process by which we could do these types of recordings, but out in the real world. And so what we did was it developed a system where we could synchronize the NeuroPace Responsive Neurostimulator with multiple wearable devices. So accelerometers, GPS, GoPro cameras, eye tracking cameras, even scalp EEG, if we have the time to do those in the future, skin conductance, heart rate, respiration, basically as many things as we could record at the same time that a person could wear. And then we had them do a navigation and memory experiment in the real world at the temporal and spatial scales of real world behaviors. So really moving around a large environment, going indoors and outdoors, and at large spatial scales. The great thing is we can also do standard laboratory experiments that have that kind of high experimental control. We can do real world ambulatory experiments with people walking around, and we're working on developing some experiments where we can present information to the participants out in the real world in very specific places and at very specific times to really try to get an understanding of patients' episodic memories for their real world experiences. And so what's important there is that memories are memories that we make and that are important to us are really formed during these freely moving behaviors and are made at the temporal and spatial scales of life. And another important fact for the work that we're doing here is that we also don't have a great understanding of how our brain is involved in parsing or chunking our continuous experience into episodes. And so one of the questions we had for this particular first experiment was, how is the brain responding, specifically areas like the hippocampus, to spatial shifts, goal shifts, or perceptual shifts that have been proposed as what we would refer to as event boundaries or event segments. And what's interesting about these is that they've all been shown to impact memory in interesting ways. The memory for the order of things that happened inside the park versus inside the city will be better than the order memory for what happened between the park and the city street. We also have increased temporal distance estimates and overestimations of the duration of the experience. If you have more event boundaries, you may remember during the pandemic, you had this kind of weird sense of time. That was probably related to the not having the typical kind of sets of event boundaries that you would typically have because you were stuck at home. You weren't getting out of the house, going to drive, and then getting to work. The extent of it was you're getting on your computer, and that is not as strong of an event boundary as perhaps moving throughout the real world. And you also have better memory for items or information that was presented around that event boundary. So in terms of investigating neural representations of memory in space in moving humans, much of what we know comes from studies of moving animals. So we do have a great understanding of the types of cells that fire, from place cells, grid cells, border cells, head direction cells, and so forth, as people or as animals move through an open field in these environments. And we also have the Rizzolatti work on mirror neurons and social place cells in bats. And there's even some work from our lab that I'll show you in a moment that talks about how you can see some of this evidence of boundary detection when you look at when you think about where you're moving or are tracking where someone else is moving, in terms of the increases in theta oscillations, which are thought to entrain place cell firing as an animal moves through an environment, known as theta phase precession. We also know that if you have an animal that's got brain recordings from their hippocampus, that you can see cells that fire at each of the doorways of a multi-compartment environment. So these are like place cells, except they fire it at specific environmental features like doorways where the space constrains itself. Or you can even see higher firing rates around the midpoint between an active part of a box and a rest part of a experimental box. So this all sort of points to the fact that there are there are neurons that are tuned to this type of information, this type of spatial boundaries in animal brains. And in human brains, we think about this with respect to having people watch videos of like everyday actions, and where you have people watch these videos and they hit a button with a very naive or kind of open-ended instruction, which is press the spacebar when one meaningful unit ends and another begins. And we let the participant themselves tell us what a meaningful unit is. They're naive to exactly what we mean by that. And so you can have people hit the button at those particular periods of time. And what you find is that there are relationships in terms of when people tend to feel like their one meaningful unit ends and another begins. It's not random. There is some structure to it. And when you take that structure and you align it to a group of participants that are being scanned as they watch an Alfred Hitchcock film, in the case of the CAM-SCAN study, or a Forrest Gump film, and you align where people agreed that there was a meaningful unit ending, then we call that the high salience condition. Say 15 out of 20 people rated that moment of time in these films as a meaningful unit ending. If you align the data to that, you see increases in hippocampal activity at and around those sort of event boundaries, that is parametrically modulated by how many people agreed that there was some sort of meaningful unit ending at that period of time. We also know that boundaries are important for things like theta oscillations. And so if you're moving through yourself through a instrumented room, as you get closer to the walls, we see this increase in theta power that is really consistent across participants. And this is even the case if you have someone else if you're watching someone else move through the environment and you're tracking where they are. But we see this activity in this kind of lower frequencies from 4 to about 15 or 16 hertz. And you see this very consistently across patients, and it's much stronger when you're on the near the boundaries of an environment. And we wanted to ask the question, to some extent, what happens if you were to walk through the wall, like in the case of a doorway? So if you were to go through that doorway there. So the way that we think about this is in terms of the temporal context model of memory. And that is that as you're going through your continuous experiences, there are moments of time where you get the faster drift in your spatial temporal context. And at those periods of time, we'd expect there to be changes in brain activity that are associated with making event boundaries and moving on from one event to the next. And so these types of events could be cognitive shifts, like shifting what you're thinking about, whether you're thinking about where you're going and then switching to thinking about what you wanted to get for lunch or something like that, to making turns throughout the environment where the actual physical context changes, or going from room to room or down a hallway, or going from indoors to outdoors, which I actually think is one of the biggest kind of contextual changes that we go through on a day-to-day basis. You can actually almost feel something different when you walk outside. And so what we were predicting in this case is that theta activity, given that it increases around spatial boundaries, that perhaps it would actually increase its activity around all kinds of boundaries, from sort of cognitive boundaries to turns to doorways. And we think the hippocampus is likely very much involved in this, but that it's part of a posterior medial network that's been proposed that allows you to kind of stitch together the memories as you're having a particular experience to become a long-term representation over time. So this required a lot of technical work. We have Uroš Topalovic has a wonderful paper in Neuron that shows how we did this in the instrumented room, and talks about how we get all of these different sources of information synchronized down to 1 to 10 milliseconds of precision, which is wonderful, and that really allows you to figure out a lot about how the brain is responding to what the participant is doing. For our task, we had to develop a system that was completely untethered from any particular environment, but could be worn by the participant. So this was movement tracking, GoPro cameras on the patient's chest, of course the neuro recording system, and this is all kind of synchronized with the Raspberry Pi. And we can also have things like psychophysiology, eye tracking, and a bunch of different data like GPS, audio information. And of course, we were doing this work in LA, so we had to have a clapper board. We also have a 360 camera that get everything that's going on around us, and then a couple monitors on iPads that tell us like how the recordings are going, basically make sure that the recordings are still working. So what do we have the participants do? So this is one of the more simple tasks you'll see out there. All we had the participants do is what you would do in some animal studies, which is have the person walk a maze around campus. And if you've ever been on UCLA's campus, you'll know that this is quite a maze. And so all we did was ask them to be able to remember the route the best they can, to be able to navigate from the end back to the beginning. And so we did an encoding walk where that was their instruction. They were just supposed to just pay attention to the route well enough to be able to navigate from the end back to the beginning. They had no other orienting instructions. So in this case, I was just sort of pointing and kind of telling them where to go as I walked next to them. There are also landmarks along the route. They would incidentally kind of see these landmarks and use them, and we have eye tracking to tell when they looked at those. And then they would navigate the route themselves 6 to 7 times. And this is where they just do the best they can. We would let them get lost sometimes so we could look at the behavior of like that moment of time where you're like, oh man, where am I supposed to go? We also did some control walks because we know things like movement speed really can affect the signals that we're interested in. So in some cases, we actually had the participants, we held the door open for them and we would have them stop in every sort of doorway. And then we had another walk where we had them walk through every doorway in a continuous speed. And in the final walk, we had them go from the end back to the beginning. So they actually ended up on the 7th or 8th walk, going from the end, walking back to the beginning to sort of make this route more difficult again after they've already learned it. We also did a landmark recognition task where we showed them 150 landmarks that they could have seen on the route, and then 150 landmarks that weren't on the route, and they had to distinguish between those. And then we've also done event segmentation in a large group of undergraduate participants where they watch the videos that are coming from the patient's GoPro camera and hitting the spacebar when one meaningful unit ends and another begins, so that we can align the data to those moments of time where there's high agreement as well. So this is one of our patients where you can see their data scrolling across the screen. And she's like completely outfitted with all of the different sensors that we're wearing. This is some of the data from her perspective. This is the eye tracking data where we're trying to calibrate the system and do synchronization of everything. And then you can see this is the eye tracking data. You can see she's kind of paying attention to where I'm pointing, making sure she's not going to trip over the stairs, trying to figure out where the actual like doorway entrance is. Some of this data was collected during the pandemic, so we had to unlock some of the doors. And so in this data, we've collected data from 5 participants so far, and we're going to be doing more of this work at the University of Utah this coming summer. And we've got about 28.5 miles of human hippocampal real-world recordings. It's about 13 hours of data. So there's a ton of data here, and we're slowly able to kind of work through all of this. One of the things we've had to develop for this is kind of a process of doing kind of behavioral coding or environmental coding and annotation. And so this is one of our GUIs that we've built that allows us to synchronize the brain data down to the millisecond with certain behaviors and really annotate what's happening, like when they went through a doorway or when they seem to get lost, things like that. And I'll show you some data aligned around those moments of time in a moment. You also might be thinking, man, this is a really uncontrolled study. How are they ever going to figure anything out about what's happening here? Well, one thing is we just tried to measure as much as we could. And we're trying to use things like through these measurements and computer vision and machine learning to try to get a handle on what's happening in this data. So in this case, we're feeding in each frame of the video into what's called YOLO version 5 that allows you to kind of parse all of the different features of the environment. And then we can also use computer vision plus large language models to try to get an understanding of the different contexts that people are in, defined more by a computer rather than by a human, to take out some of the sort of subjectivity. And we're doing this with our collaborator, Luis Garcia, in the computer science program here at the University of Utah. And so we're also, of course, doing the brain recordings as they're doing all of this stuff. And in this case, what I'm showing you here is a power spectrogram where brighter colors are indicative of higher power or larger amplitude signals in a particular frequency band at given moments in time throughout the task. And so you see gray bars indicate when people were indoors, green indicate when people were outdoors. And one of the things that really just jumped off the page, even when we were doing just the raw recordings as people were walking around, is that we tended to see stronger activity in this theta alpha range when people were navigating outdoors rather than indoors, which is something that hasn't been described before. So we're really excited to see something like that, these big contextual changes. And we're trying to understand what those really mean. We can also align the data around, say, spatial transition. So each of these black lines indicates a spatial transition that happened at particular moments throughout the task. And we can align the data around those moments of time as well. So first, as I mentioned, this sort of stronger activity when people were outdoors than indoors really just kind of jumped off the page. And so we looked at that in the anterior hippocampus. We've got every patient that we have, all 5 patients have coverage in the anterior hippocampus. 2 of our patients have coverage in the lateral temporal cortex. But as you can see, in both cases, you see this kind of increase in theta power, but different kind of characteristic theta peaks in this case. And when we look at across all of our participants, across all of the channels that we've collected data on, it's about 20 different channels of data that we see this activity primarily increase in this kind of 5 to 8 hertz region, which is really standard kind of high theta activity. And you can see this across almost every electrode, not just the hippocampal electrodes, but even lateral temporal cortex, pararhinal and entorhinal electrodes, and so forth. Even posterior hippocampus shows this type of effect. And so one of the first questions we wanted to ask was, how much is velocity really, is it just that they were moving faster when they were outdoors? Or is it in fact that there's something else about them being outdoors that really mattered? So we fed this data into a generalized linear mixed effects model for this 5 to 8 hertz power to see what factors might explain, be significant predictors of these changes in power. And we show that even when you control for how fast they're moving, in whether you're indoors or outdoors is still a significant predictor of power. And for velocity, when you control for whether someone's indoors or outdoors, velocity is also a significant predictor of power. And between these two, we explain up to 87% of the variance in this 5 to 8 hertz theta signal. So there's a lot that we still want to understand here. So a few more final slides. What we see here is when we look at, I'll show you just data from only the anterior hippocampus, since all 5 of our patients had coverage there. And what we're doing here is we're subtracting out this kind of 6-second baseline that happens before a particular event. In this case, these are spatial transitions, like a doorway, going up a few stairs, anything where you need going from a sidewalk to a parking lot, anything where you need to kind of shift what you're paying attention to. And we're subtracting the data from that baseline, or we're permuting that data from the baseline period and then comparing the data that happens around this event. And what we see is that as soon as you go through some sort of spatial transition, like these kind of general spatial transitions, you start to see these increases in theta activity. This black line here is their average velocity at any given moment in time. And so as you can see, you actually get the opposite of what you would expect. You get an increase, even though they're slowing down here, an increase in theta power, even though they're slowing down. So this is really exciting and suggests that, some of our ideas about event boundaries or sort of spatial boundaries and how they might affect hippocampal activity seem like they're being supported by this data. You can also look and see very different types of patterns of activity when you do the same type of comparison. And what you see is right before they go through the transition, relative to this baseline period, you see this increase in theta activity that happens right before a large, lower frequency decrease in activity as people are making a turn. So this is right at the point where they're starting to change the angle of their body relatively to turn around a corner in the environment. We can also see this even at choice points. So when you either go straight or you make a turn, you see this type of activity. And what's another interesting tidbit is that this activity gets stronger as the person becomes more familiar with the route, this sort of anticipatory activity that happens before the transition, which is really exciting and suggests that there's some sort of learning happening as they anticipate making these turns. And then finally, we show that we have some evidence that when someone starts to show the behavior of being lost as they're walking around, and you compare it to this 6-second baseline period, you start to show these decreases in theta activity as soon as they are like noticeably lost. And you see these increases in gamma activity. And this would be a characteristic kind of memory signal that you would see in a typical laboratory experiment, known as like a spectral tilt, where the higher frequencies increase in their frequency or in their power, and the lower frequencies decrease in their power relative to the baseline. And so finally, I just want to leave with this message, which is that, we totally recognize and feel like it's really necessary to do these types of observational studies where we have some hypotheses, but really we're trying to observe real world human behavior alongside neural behavior. And I think I want to kind of deliver the message that these new tools offer a new observational perspective on sort of theoretical questions that we have. And these new tools include all of the different technologies that we've used in these studies. And we really need to be making the effort to try to figure out how to use these types of new tools to get out of the laboratory and into the real world. But one issue is that, currently hypothesis testing is the primarily incentivized factor in funding decisions and publication decisions. And I think that if we incentivize observational or exploratory studies of real world behaviors and complex tasks, that will allow for better informed hypothesis testing. If we go back to the original place cell studies that O'Keefe and Dostrowski did, they had kind of these new tools, these new glass electrodes that allow you to record from the hippocampus, and these kind of lightweight wireless recordings that you can have on a rat as they moved around in the early 70s. And they looked at 20 or so different behaviors to see what cells were firing to what behaviors when they moved the animal through the environment, or when the animal was grooming or turning their head or whatever it may be. And they happened to discover something that eventually was awarded a Nobel Prize in place cells. So, these sorts of insights and these sorts of naturalistic studies have already demonstrated these really incredible, abilities to discover new things. And that with new technology, we should be taking these observational perspectives and starting with the first step of the scientific method, and then getting to trying to figure out and nail down what some of those relationships mean with more controlled studies and hypothesis testing. So in conclusion, there's a need to build these naturalistic paradigms that can assess memory and neural function in real world settings if we ever hope to realize the promise of things like neuromodulation. That capturing real world hippocampal recordings and stimulation in tandem with wearable devices is a first step to understanding memory in a large scale real world settings. And finally, that future studies using augmented reality in real world settings may allow us to test whether we can enhance memory for meaningful events in patients' lives. And so we're working on all of these sorts of questions and are excited to continue to work on this. So I'd like to thank you for your attention, and thank you for attending this talk. If you have any questions, feel free to reach out to me via my email or through our lab website. Thank you so much.